There's nothing out there. The first thing I tried was something called a hairbrush. If you know what a floating microelectrode array is, it's just a bunch of wires that are all glued together at the backside, and then you stick the wires in. It's one thing, but it's just a bunch of wires. And that takes a lot of effort for people to build. And this is all research lab sort of things. So grad students don't make good technicians. So it's like they had to come up with a better idea. And Pat Campbell, this is Pat Campbell right here, this is Kelly Jones. Pat Campbell said, hey, if we just use a highly conductive silicon, it carries current, will work just fine. And that's how the impetus for this thing came out. They are both referenced as kind of the source of the Utah Array. There's a slight difference. So how do you make a Utah Array? This becomes important because it distinguishes them. So this is a piece of silicon. It's a millimeter this way, and it actually goes that way and that way. It's a disc. The disc is flat like this, one side on the other side. First thing you do is you make a little notch on this side. Use it something called a dicing saw. This is all IC manufacturing stuff. Why you do that? So you can fill it with insulating glass, which will become apparent why you need that later. And then you take that same dicing saw and you cut down this way to make individual shafts. You cut it this way, and you also cut it this way. If you were coming and look at it from this side, it'd be a little rectangular pillar, about 100 microns by 100 microns, and then a millimeter high. And then use some fancy chemical etching. This is where silicon helps you because it's got certain crystal structures that you can take advantage of to get a nice sharp tip. Now you've got the shafts. Silicon is a terrible, terrible electrode. So what do? You put metal on the tip. Could be, originally it was platinum, now it's platinum or rubidium oxide. There's nothing that says you can't put anything else on. And then you put the metal on the other side, this backside, that's where you're gonna put your wires to connect. And then you insulate the whole thing. We started with polyimide. We tried silicon nitride, silicon oxide, ended up with parylene C, and silicon carbide is an interesting idea. And then you etch just the tips off. So how do you do that? How do you just get the tips etched off? You end up poking this thing through a piece of foil and bring the etching stuff on this side. It doesn't get down to the insulation back there. And that's how you get it. The distinction between Dr. Campbell and Kelly was trying to build diodes between the glass insulation. They were actually using IC manufacturing ideas to make a diode so the electrodes were actually electrically isolated. Good idea, didn't work out. It would have really been better because it's one less manufacturing step. Now, this is actually the real magic. This is why the Utah Electrode Array exists today. This is Vin Nago. Vin was the technician back in Dick Norman's lab. He was there before I got there in 94. And if we needed an array, we went over to the other side of the lab and said, hey, Vin, I need an array. He said, well, when do you need it by? And he says, no, that's too quick. I can get it by this date. And how good does it need to be? That's how we got our electrode arrays. He was the one who did all that preceding slide, did all those steps, and made really wonderful arrays. And he was the poor guy who went those little tiny dots in the backside and soldered it to a connector. When it was commercialized, which I'll talk about in a bit, he was their very first hire because he was the man. He knew more about making Utah arrays than anyone in the world. He is still at what eventually became, the company eventually became, which is BlackRock. It was paid really, really well. And this is actually the company headshot of him. I was there yesterday and asked for it. They gave it to me. But this is the real magic. So now I'm going to start talking about individual groups. And here's where I have to apologize. I'm talking about a lot of stuff I ran into. Mitch over here, unfortunately, is in the Greg Clark era. And he's not on a slide. I apologize. But these are all the people I knew back in the day that eventually got through. This is Dick Norman over there. And this is like a minor subset of Dick Norman's graduate students. But these are the various tasks. I'm not going to talk about every one of those. I'm going to talk about what I think are the important ones. The first one, Pat Lachey down here in the corner, much, much later picture, gray hair. How do you get this array in? You can't just push it in. It's 100 shafts There's 100 shafts. You try to push it into cortex, and all the cortex does is dimple out of the way, which is not good for cortex. Two ways you can do it. You can very slowly push it in. And neuroscience, study for neuroscience meeting like 10 years ago, that was like the thing everyone was talking about, slow implantation. And people like take hours to implant. I'm sorry, I'm an engineer. I want to get to my experiment. I don't want to spend a lot of time watching this thing going in. The other way is fast. So if you shoot it in, this inserter over here, pneumatic inserter, is what Pat Roche is famous for. He's probably the most, his paper about the inserter is probably the most cited Norman Lab paper because people always talked about the fact that they use this thing. It is still in use 30 plus years later. So he came up with the idea of pneumatic. He tried a couple other ideas that didn't work as well. So this is big important piece. Second big important piece. Remember I talked about exposing the tips of the shafts, tips of the electrode to the metal? You can either make a very unexposed tip or a lot of exposed metal. What's the right answer? How much? Just depends how far you push the foil down. Craig Nordhausem, which is also an old person picture, looked into this. What he found was about 30 microns to 120 microns is a good range based upon that slide over the corner, which is showing the signal noise ratio as a function of tip exposure, how much. And it said this stuff down here is better. This space over here is empty, so don't make it big. And also he looked at the impedance as a function of how much material is exposed and thought this was a better. These are platinum, and this is where most of the data are now. BlackRock will quote 50 for you. They can do 30 if you ask nicely. They can do 100 or 75 if you ask nicely too. The other thing that Craig looked into is connectors. This is a beautiful little device. The initial experiments they did, they put it in, and then they brought this little probe thing with one little needle on it and very carefully touched the back of the array where that metal was and recorded. And then they picked it up and went to the next one and recorded. That takes a long time. What you really want is a connector, right? So Craig was also involved in the connectors, and he was the one who came up with the idea, how do you do the soldering? And by the way, that's technology is not used anymore. Little divergence here. This was my Society for Neuroscience poster in 1996. And Jake's got a smile and says, a couple of people recognize this. What the heck? That's not a poster. This is how we did it in those days. We used PowerPoint and made a whole bunch of 8.5 by 11 pieces of paper and then glued them to pretty cardboard and then put the cardboard up. Yeah, that's how we did it. Because that was the technology at the time. So I just thought you guys should know about that. However, what we always found was the following. Here we are, here's our beautiful poster. We want to show our great science, the wonderful insights we had, the analysis we did, and everyone go, where can I get that? I'm not kidding. That was neuroscience for many years for us. Where can I get that? And that thing was, well, I go over and ask Ben. So because of that, Dick decided, let's commercialize this. So he started Bionic Technologies back in 1996, according to the state of Utah, working with Brian Hatt, who was an entrepreneur back before the word existed, to commercialize this thing. They commercialized the array so they could actually sell it. They also created recording hardware because you've got an array, you need recording hardware. In those days, people were used to using a couple of electrodes and they had like a 2-channel amplifier. This has got 100 electrodes. That ain't going to do any good. So you need something that can actually work. They also made many manufacturing processes improvements because things you can do in a company that just don't make sense in a research laboratory. In 2010, Cyberkinetics merged with them, which really they bought them out. Cyberkinetics was a group out of Brown University, mostly run by John Donahue. They wanted to go into humans, and they didn't like the fact that Bionic Technologies was kind of slow in delivering rays, and they weren't high quality. So they wanted to take control of it so they could actually do their work. This is where the source of BrainGate, hopefully you guys know BrainGate, came from, with Cyberkinetics. Somewhere in the late 2000s, in the aughts, they realized that this device, as cool as it is really not a big moneymaker. What you need is a medical product. So they're working on some medical product, which I don't know what it was. And they eventually gave up because the FDA basically kept telling him, no, you need to do blank. So they never got FDA clearance. They ran out of investor money. So BlackRock bought them out. And now BlackRock owns the company. And still does. And still makes everything. So that was a segue. And another interesting segue was back in the day, the first year that they had a booth, they went to the Society for Neuroscience meeting, had a booth. And guess who the booth babes were? All of the grad students, like myself. And Alma Bronner, who you see a little bit later, was the booth babe. And we got paid pizza and beer, and we were happy. So hopefully you guys get a chance to get in a startup company like that. Okay, getting back to the important stuff. So another thing is, how do you translate this into chronic application? Pat Rocher, the fellow who had the inserter, was doing auditory cortex, implanting those. Our connector at that point was a 12-pin connector in a titanium pedestal. 12 pins, you get 12 of your, or you actually get 11 of your electrodes connected to it and a reference, and the pedestal itself acts like the ground. And he was finding that the bone would grow into the array and actually extract it. There's one of his histology pictures where it's literally an array encased in bone. So how do we make this thing stay where it's supposed to be? Ed Maynard down here in the corner came up with this basic idea here, which is you implant, you put a piece of Teflon over the array under the dur, you suture the dur back together again, and you put preclude, which is it's a Teflon thing made by Gore. Craig Nordhausen worked for Gore at the time, or I suspect still does. So hence he was providing us with stuff. And then you close it off. And this actually was the magic that allowed you to go into chronic implantation of this device and not have either the tissue extrude it or the bones just yank it out. It's still, the basic idea is still used today. For example, this is what Eduardo's doing in Alicante. The other thing he did was he actually is the person who got John Donahue interested in the Utah Array. At that time, Utah had no people doing survival non-human primate work. There's a lot of acute work being done, non-survival, but no survival work being done. However, John Donahue at Brown University was doing this, but he would make a little port and then he'd bring the monkey up and they'd stick a new needle in every day, a new electrode in every day. Ed, Dick Norman, and Donahue kind of got work together, and Ed ended up going over to Brown, working on the implants, working with the monkeys, and getting results. And the important thing he found, merely a very preliminary point, is this slide over here is trying to show, if you look at individual cells firing over there, they basically only speak to a little. Over here, when you start looking at the second order model, you look at the correlation between the firing, you get a much richer information set. Very simple thing, but it shows that you need lots of electrodes to make decoding work. That was Ed's. Shane Guillory. Shane actually started there before I did. He was an electronicer. He was the best analog digital person I knew who actually was a grad student, not in an EE department. So every time we needed something, Shane made it. So the amplifier we're using for those early things, Shane made it. We used to have it. It was a little plexiglass box lined with foil for noise. Great guy. When we started working with the Brown University, John Donahue's group, the NIH said, oh, but they need a high channel count data acquisition system, and they didn't exist at the time. So the NIH actually paid Utah, Shane, primarily, to create that device and the software associated with it. What came out of it was something called the NSAS, Neural Signal Acquisition System, which is the predecessor to what's currently the Cerebus. The NSAS only recorded spiking events. It didn't record continuous data. He developed this stuff. Bionic Technologies was very interested in it, so they basically brought Shane on before he finished his PhD, and Shane to this day has yet to finish his PhD. And I think he's no longer capable of doing it. He also got tired of, when he got, Bionic Technologies got bought out by Cyberkinetics, he really got unhappy with the company because they were being very slow in updating the hardware. So he formed Ripple, which came out with a better hardware, cheaper hardware. And he's no longer Ripple. I think he's working Shy Show him. Also, Almond Bronner. This is our booth babe, a German gal. She wanted to move into peripheral nerve. I think Dick really wanted to move into peripheral nerve, and all happened to wander in at the right time. So how do we take the Utah array and adapt it for peripheral nerve? Well, if you take a Utah array that looks like this is the nerve. Those little gray circles are fascicles. The white stuff is connective tissue. And you want to have the electrodes in the fascicles. You take a flat array, you're just going to get a whole bunch of points at this depth. That fascicle's not going to have any, that one's not. And then you get a lot of repetition for the other guys here. Bad idea. But there's nothing that says you can't take your array and make the shafts different lengths. And then you get something like this, and then all these little spots here represent electrodes you can actually get signals on or evoke activity. So this is how you get a good sample of it. By the way, this is a lie. The vertical space is 100 microns, the horizontal space is 400 microns, but that grid is square. So it's actually squished. That being said, I've never ran into in my entire life a round nerve. They're usually flat. This part here, she compared it to a cuff. These little hashed in are how much current does it take to evoke 50% of maximal force using the array, Utah array, and then a cuff being wrapped around the nerve, stimulating the exterior of the nerve, and you get a lot lower current. Also, you get greater selectivity. It's not shown here. She also worked on some connector systems, early ideas that kind of worked. This is a little hard to interpret. So this is the 10 by 10 Utah array. 10 high and 10 across, and each one of the little boxes represents an electrode of the Utah array. The letters in here, for example, this T stands for tibialis anterior. Hence, when that electrode, this current flows through it, it evokes that movement of the tibialis anterior. This is really beautiful because you notice also the coloring in the background represents different fascicles. It's not like these things are randomly organized. It's all nicely organized. So if this electrode doesn't do a good job of tibialis anterior, its neighbor might.
Dave, was it cat or rat that she was doing? Was it cat or rat?
This is cat. You could not put a 10 by 10 Utah array in a cat and rat. Well, you could, but do a lot of electrodes, not doing much. The other thing is here's the currents that allow you to get there. What was the threshold current? So this was really important to us getting into peripheral nerve, which someone here might care about. Alma, when she graduated, went and worked for Bionic Technologies, then cyberkinetics, and then she decided she really wanted to be an academic and returned to I'm getting better at names. I'm getting old. A university. Another thing, since now we've talked about cortex, we've talked about peripheral nerve, there's kind of an intermediate zone, which is what's a cranial nerve? Is it peripheral nerve? Is it central nervous system? Who is it? And the short answer is it depends on what nerve. Weirdly. So I assume you know what a cochlear implant is, the most commercial successful stimulating device in the neural systems. The problem with the cochlear implant is you put the electrodes in the cochlea, the neurons you're trying to excite are separated from the electrodes by a bony wall, admittedly porous, but still a bony wall. So you're not really very close to where you want to stimulate. So it takes a lot of current to actually evoke neural activity. The other thing is getting into the apex part is difficult. So then try it, and the apex part has all your low sounds. So it turns out that's really important for interpretation. So what happens to the cochlea? Well, all the neurons of the cochlea go through something called the auditory nerve or eighth cranial nerve, and then they go to the brainstem, and there's the nucleus right there. The thing is probably all about that long, but it's in a nice secure location. That is an alternative to exciting the cochlea. Why don't you just excite the neurons in the eighth cranial nerve? That was the idea here. So this is a slice. This is the cochlea. This is like the circles coming around, and this is where you're going to put the array, and this is where all the fibers coming from the spiral ganglia, and they're heading to the brainstem. So let's put the array right there. It's smaller. You can't put a 10 by 10 there, but you can put a number of them. So Arun Bhatti from India, he was actually an ophthalmologist in India and came to do his PhD here. Because of that, he was a really good surgeon, And he was followed on by Sunjae Kim later. we just extended this. What do they find? Well, this is what's called an EABR, auditory brainstem response. When you hear things, you go, when you're a little kid, when you're a baby, they test your hearing and they just basically blast a sound and they record stuff and go, yeah, this kid can hear. If you were to go to an audiologist, they might do this having problems hearing. And basically, the idea is as you increase the stimulation current, the bumps in here get bigger and bigger until it eventually saturates. So this is increases in current, and somewhere down here is the threshold. This is just a sign that says all the wiring's working. Except for it only says the wiring works till it gets to the brainstem. Soon-Jae Kim extended this idea, which is we're stimulating the auditory nerve. Where does it go? Auditory cortex. Crosses over. So why don't we put an array in auditory cortex? We've been doing that for years, and put an array in auditory nerve, or the eighth cranial nerve. We've been doing that a little bit longer. And we stimulate here, we should be able to see activity here. That's what he did. And he found out that's the case. So these two boxes represent two different electrodes on the array in the auditory nerve. And this is the growth curves for various channels of the Utah Electrode Array. So it indicates it's actually getting the brain, it's doing something in brain. Now, this doesn't mean that I stimulate the one over here on this side and I'll hear a train whistle or something like that, but at least indicates that the information is getting across. This will become important later. So that, I think, so we got all those bits and pieces. Now I'm making a huge jump in time because there's just so much to talk about. So Eduardo Fernandez here, a very old picture of Eduardo Fernandez. This is probably from when he came to Utah in the 1990s. I was actually there at the time as a student. He's on a couple of my papers. And he was interested in the vision system. And he came to do some research. He was a faculty member at the time, young faculty, with Helga Kolb at the University of Utah, who's a very well-known, very famous person who talks about the architecture of the retina and the neural architecture and signaling, things like that. He's going to work with Helga. Well, it turns out Helga is married to Dick Norman, and they're staying at Helga and Dick's house in the Harvard Yale area. And he gets talking to Dick. And Dick's talking about this vision prosthesis and all the cool stuff that's going on. And Eduardo says, no, I'd rather do my rotation. Dick Norman. So he ended up rotating to work with Dick. And he's been part of that group ever since. So a much older picture, which I don't have, is he's actually now taken that vision prosthetic artificial eye project, which talked about 30 plus years ago. He's doing this in Alicante, Spain. He's implanting people with Utah arrays and having them walk. And he's got this nice little room. I was over there a couple of years ago. This is like a street in Alicante, but it's inside of a lab. And they can actually change the street, move things around. And people with prosthetics, or vision prostheses, are actually walking around here and sitting down and reading things and doing all sorts of cool stuff. So that actually reached humans. He's continuing to do that work. Last time I talked to him, which is some meeting I went to, NIC, maybe, EMBC, maybe, it was NIC, that he's moving forward. And he wants to start getting dual implants and things like that because the 10 aren't adequate. He wants to go further. So all that work finally has reached humans. Also, oops, you're back.
How does it compare to second sight?
Also,
How did it compare to second sight?
Different. So everything now is still external. Second Sight actually had a fully integrated stimulation system that people are reporting it as valuable. I think also people report Second Sight as valuable, the retinal approach. Second Sight doesn't exist anymore. And I you know, to be honest, so far, I haven't heard any discussion about commercializing this into a vision prosthesis. I think, you know, the first slide about the artificial eye, there's still lots of bits and pieces. He wants to go wireless. Anyone want to do wireless? There's lots of things he wants to do, which 30 years ago we discovered were not as easy as they look. So, yes. I also talked about the auditory nerve implant as an alternative to cochlear implants. Done back in the early 2000s. Whatever happened to that thing? Well, it's been talked about for years. After Sunjae and Kim graduation, the funding kind of dried up and we couldn't get any more money for it, but it was an interesting idea. And Florian Sulzbacher kind of kept that alive. Talked to Hubert Lim at Minnesota, who got an NIH grant, a UG3, UH3. UG is a 3-year development part. If you get through that, you go to the UH3 part, which is 2 years in humans. It's intended for a clinical application. It's also intended for a commercial device, prototyping type idea. Hubert got the UG3 part. He needed someone to do the cat work. It was supposed to be done at Feinstein. Lauren Reith was at Feinstein at the time, but Lauren actually left Feinstein and went to West Virginia. And the person that was going to do the cat work actually went somewhere else. And suddenly, like, we have no cat work person. They're doing also non-human primate work. They're going to do both. The cat work is mostly to prove that you can do long-term recording and simulation and for safety purposes. They were looking for someone who knew auditory nerve and cat work. Okay, I don't know auditory nerve, but I was in the room at the time. So, and Lauren knows me well. So they came back to me and we, myself, Richard Gale, who's our magic, he's our ear, nose, and throat doctor, a guy at the hospital who does our implants for us. Mitch, who some of you might know, and Holly Holman, who you know, hopefully, did this. And actually, we were very successful. No one had ever done a chronic implant in auditory nerve. We got through chronic. We proved it works for months and months. We came up with ideas to make it work better. That's where Holly came in, as we did a second study where we actually stimulated it chronically to show that stimulation is not going to cause problems. And we were successful. And this is what got the UH3 part of the program funded. Currently, they just started. In fact, it's still kind of in that weird spot because, as you may be aware, the federal government's a little strange right now. But we got the notice award for the human implant work. It's going to be done in Hanover, but it's a fully integrated system. It's got a Med-L stimulator, a Utah Array in the end of it being stimulated. It's all implanted. And they'll have this kind of standard sort of cochlear implant sort of stuff. It's just that we're using Utah Array instead of a cochlear implant. The non-human primate work, crash and burn. They have a really weird inner ear structure that just made implanting Utah Array impossible. So this is one of the things I'm really proud of. So we actually managed to get the artificial ear, the artificial eye going, and we're getting the artificial ear going too. So that's ginormous. Ken Horch. Ken Horch was the other half of that program. Keechel Shah was Ken Horch, the guy with the pixelized vision. Ken's actually is an interesting case. Everyone hates Ken. I don't know why. He can be a rather rough person, but he's actually a very good person. And usually he's right. Over and over again, I've had disagreements with him. He's been right and I've been wrong. I accept that. He was here at Utah at the time. He had this thing called the life electrode, longitudinal and intervesicular electrode. He actually had it well before the Utah Array, and he was actually doing neural interfacing before I said the beginning of neural interfacing. Sorry. He also did a whole bunch of stuff, and I'm only going to hit a couple of them. So first I'm going to describe the life. It's like a little idea. If you take a really skinny piece of wire that's insulated, I mean, I'm talking tens, fives of microns, it's insulated, and put a needle on the end of it, and scrape a little part of the insulation off. And you take that needle and thread it into a nerve and pull it through and get that part that's exposed inside the fascicle, inside the nerve, and then cut the needle off because you don't like to leave needles in people, and then bring the other end out back to your recording hardware. Simple, easy, great idea. Even better is they realized later the wire is still pretty stiff. What if you metallized like a really fine fiber, take a piece of suture. Don't use the polyamide type, use a much more finer type, and metallize the outside of it and then insulate it and expose a little bit of it and put a needle into it. Great idea. And you can put a whole bunch of these in by just threading them through one at a time. So that's what they came out with. It's got great mechanical, biocompatibility. And importantly, when they went to do humans, they asked the FDA, and because literally you put it in, and when you're done, you take that wire, pull it out. And that's it. There's no second surgery, none of that, none of the above. And if it's, they got a problem, pull it out, wash it off, and sorry, go home, guy. So they decided it's a non-significant risk. There's actually a letter from the FDA, fairly old at this point, saying that these are non-significant risks and that you can go ahead and do clinical trials with it. So that's why this is so useful. One of the first people involved was this was Ken Yoshida right here, one of Orchid students. And he was looking at, well, one of them is great. What if you put two of them in? Let's say you've got a nerve, it's got a fascicle, and you manage to get two of these things into the same fascicle. Is it going to be better than one? The answer is yes. With the answer, what we found out is if you stimulate things, so these little arrows here, you stimulate one fiber or the other, so the up and down arrows here are doing it individually. This is the pulse width modulation. This is the force you produce. That's down here. And if you do some crazy things like you stimulate at the same time or other things, that it gets bigger, and if you have to stimulate them at different times, so they're not interfering with the other. Remember refractory period? You don't want to stimulate one in the refractory period of the other. If you get them far apart, you can get really high forces out of them. So two is better than one, clearly. Why is it not causing, why can you stimulate them independently? Well, you've motor units, you're exciting one motor unit that goes to a nerve or muscle and a different motor unit that goes to the same muscle. So you're not actually exciting the same part of the muscle simultaneously. So it avoids issues like fatigue, which I'm showing here, which if you do two different electrodes, stimulate them, this is the force produced, and then this is the how long they stimulated. The fatigue sets in much later, out of a minute, versus if you do this simultaneously, individually, it drops off earlier. So this is an idea, and interestingly enough, the Norman group stole this idea and has been using it since then. Other thing he did, really cool, way back in the day, is, sorry, this thing's here. This is a stimulation setup, and he's just got a life electrode he's using to stimulate, and he takes another life electrode and uses to get sensory feedback. You got proprioceptors, right? So I can use proprioception to tell you where the joint's at, and I can stimulate, move the joint, and he did closed loop control back in the early 90s of that. And amazingly enough, Horse Group doesn't get a lot of reference for that. Other thing he did was he actually did humans. He had a non-significant risk. Dylan, everyone calls him Dylan, actually did humans. Here's kind of their setup right there. They've got a really weird amplifier. I was actually partially involved in this. They hired an undergraduate to do that, and I kind of advised the undergraduate. And it was the worst amplifier design I've ever seen. Even I knew it was bad, but it worked. What do they find is that they can produce percepts. So over here, the vertical scale is what does the person say their joint's at? And this is the current they're stimulating at or the frequency they're stimulating at. And you get a linear relationship. As you increase the current, oh, the finger's flexing or whatever joint is involved. So you can give a sense of proprioception back. Other thing is that, well, you've seen some of these. Some of you have seen some of these in the Clark George paper, which is, here's my hand when I stimulate an electrode. Where does it feel? Where does this person feel it? The same idea here is that you can get individuated perception. These are 4 different subjects and different electrodes, and the regions represent different parts of the hand that the person perceives something happening from that stimulation. So it showed that basis of all this capability. 3 great papers, and then no one ever talks about them anymore. Whoops. And I can't, Ashley's not here. I also can't talk without talking about Vivian Musharwa. I've known her 30 years, and I still can't pronounce her last name. She did spinal cord stimulation and showed that the spinal cord has an organization. Each one of these regions represent a different muscle, and you can actually stimulate parts of the spinal cord to get muscle activity going, get muscle movements. This has gone on since then, and she's doing a wonderful job in Alberta. Actually, Dalrymple happens to be one of her Vivian's students, and Vivian's fairly famous. So that's the Horchians. I feel bad here. The Tresco group, they're part of the neural interfaces. So what does Pat Tresco do for a living? Histology and mitigation of bad outcomes. People think mostly about the first part and not a lot about the second parts. So Patrick Tresco has looked at, when you put an implant in, what's happening? What are the cytokines? What's the various types of markers or various types of cells are involved in this process? What happens across time? And then what can be done to help mitigate that? Are there certain drugs you can use? Minocycline for a long time was like the magic cure that if you gave them minocycline, it was going to solve all sorts of problems. Doesn't really work very well. He's been a big part of this. This list is way too small. If you go look at his publications, I need like 5 pages. He's done a lot of stuff. The important ones I want to point out, Roy Byron started looking about the biocompatible neural interfaces. Amazingly enough, no one had thought about that. These unique materials. He actually was using a NeuroNexus type probe, silicon-based also, but a lot smaller profile, and looking at what's going on. This is showing at 2 time points, 2 weeks, 4 weeks, and distance from the implant, 3 different markers for 3 different things happening. The immune response. This is a stab wound of control, which is just to stick something in, take it out, close up, and go home. And then this is leaving the device in and they're on the same vertical scale. This looks a lot worse. Something's happening. It's a function of distance versus, not much going on here. So this is the work that Roy Byron actually did. Ben Christensen, his first name is Michael, but he goes by Ben. And his publication's Michael too. It's out from here. He's up at School of Medicine. I forget what department. I think surgery. He was involved with a cat study that I did, which I'm not actually going to talk about. Here's our cat. Here's some nerve. Here's the connector that we developed. And we implanted these things. This is part of the early process of actually getting into humans, showing in a cat that this is going to work. Up to that point, they'd done some long-term recording in cats, and the answer was that the recording dies out in a week or 2. Stimulation seems to persist. Then did some histology for us. And amazingly enough, and here's just an example of this is fiber diameter, and this is just the proportion. And the contralateral is the solid line, and the either distal implant or proximal implant, things drop off. What happens is the large cells die off, and that causes the small cell proportion to be larger because you have less cells. However, his endpoint was, this is okay. It's not the end of the world. So you got us through that part. Those are the 2 key points I was involved with, or I know of out of the Tresco group. There's lots of other stuff that I was actually in this lab for a year that happened. I'm just going to bypass it. Okay, Dick Norman was getting old. And he said, what's going to happen? When I retire, is this whole thing, House of Cards, going to crash? At that point, we had 2 fairly new faculty, Greg Clark. Hopefully some of you know Greg Clark. Most of you do. And Bradley Greger. You all know Bradley, right? Yeah, you know Bradley? He's a nice guy. I like Bradley. He was a real pain in the butt during a certain publication. But so Dick had the idea. I will give the peripheral stuff to Greg, and I'll give central nervous system stuff to Bradley. Bradley did monkeys, which made a lot of sense to doing that. And he did a lot of stuff. He did acute implants with Danny McDonald and was looking at how this works. Ulmer Bronner started it. Danny basically carried it forward. Wireless stimulation, some cool stuff. Reed Harrison was a EE guy here, was doing all sorts of wireless stuff. Chronic implants, myself and Noah Ledbetter. Cat standing, Dowden, Wilder, and Hyatt. That then got us to humans. Somewhere in there was a Mitch Frankel, but hey, he doesn't count. The end point was, this is the important thing, all this work. got us to two programs, RP 2009, 2000, the mid 2000s. We're going to make a prosthetic for people with limb loss, and it's going to be available and commercially out there by 2009. It was a good idea. It came out of DARPA. It failed. They followed on two other programs, which I didn't list here. Those failed. Did succeed in the making a device, which eventually came out with the haptics program, which did get us to that point where devices were made, setups are done, and we're living off of that. A lot of you are living off of that source. So let's talk about what happened. So Noah and I basically just showed we can implant for a long period of time. This is impedance of electrodes, and yes, they last for months. This is the number of channels with neurons, last for months. Life is good. Devices work. And we actually did some decoding with it, kind of crude decoding. We actually showed that we could use the proprioceptive information to decode position. And in a paper that never got published, but that then allowed us to get into humans. This is our first group. This is not the whole team, but this is the people whose names are in the paper that are important or extremely important are Heather Work right here, Tyler Davis right there. Hopefully some of you know Tyler. And Doug Hutchinson. Hopefully some of you know Doug Hutchinson. Doug is the orthopedic surgeon who actually does all our implants for us. Dick managed to get some weird sort of seed money, and we use that non-significant risk determination to say, well, any Utah made device is not a significant risk, so it allowed us to put Utah arrays. We didn't tell the FDA. We were doing these initial patients were actually people going in for neuroma. They're amputees going in for neuroma, and we implanted at just proximal to the neuroma, and that piece of tissue was going to be cut out. And we only did it for a month. So, and no one had bad outcomes. Life is good. And the same sort of thing we did in the cast in the preceding slide. This group did with these humans, which is impedance versus time and signal noise ratio versus time. And at least for the first month, everything's cool. Also, they did a little bit of decoding. Tyler came out of Bradley Greger's group, and they used something called a Kalman filter, which actually is kind of the standard everyone uses. And it gave them this capability that we didn't have when we did the cats. And it actually did pretty good, which is here's the R value. How many movements can we actually predict? Well, this is the second subject. I mean, get pretty good predictions, even out with trying to do 13 different movements. So this has got us to the point we're doing humans. And at that point, I'm going to stop talking about the Clark group. I'm missing things like the neural interface lab, stuff like that, all sorts of stuff you guys don't know about. Bradley Greger. You all know this guy, right? Bradley did not get tenure, so he left for Arizona State. He did all sorts of non-human primate work. He actually did a lot of stuff. He looked at muscle decoding. He looked at visual cortex. Spencer Kellis, I believe, is online. BlackRock did human speech decoding. He did all sorts of clinical cool stuff. And he was also involved with this first set of human implants, a very large team. The decoding, Justin Baker, right here, and Spencer Kellis, and I was involved, but I used my stupid old decodes and that didn't work as well. What they did is they had a monkey trained, and he used his manipulando, this is his hand, and he had little sensors in there. He was trained to, if a light came on, it means flex your index finger or something like that. They implanted IMEs. These little white things are called IMEs. We didn't make them. They were given to us as part of the program. It's just a wireless EMG recording device. They kind of work. And picking up EMG from these things, we can decode what the monkey was doing. And hopefully this video is going to work. Aha! So we were actually using it to decode what the monkey was intending to do. Very simple decodes here. It's one channel. And I apologize for the really weird visualization. What's this got to do with this guy? And it was given to us as part of the RP 2009 program. So that's what we use for all our decodes. All real time, by the way. Tyler Davis again and Kian did the visual cortex implants, which were really fundamental to getting to the point where Eduardo could do them In humans, these are all done in non-human primates. The important thing is here. So he trained a monkey to fixate here, and then you would put a spot of light there, and he's supposed to saccade to that position. You have an eye tracker, so you know if he goes there. And then you have another spot out here, another spot here, etc. And the monkey was trained to do this. But now this guy's got Utah arrays in visual cortex. What happens when you stimulate? So he fixates here. And this is where your array is. This grid, green, red, and blue grids are where they were stimulating in the visual space, because they know where it is in the visual space. The arrays are in visual space, and says they should saccade to those locations. And lo and behold, that this is where he ended up. The monkey ended up. Supposed to be there, but he did something. And the circles is kind of the arrow bars sort of idea. So monkey was saccading at the right spots. This is part of the reason why, hey, it's not just like you fire up a phosphine and the monkey doesn't care. It's randomly appearing in space or whatever. No, it's in the same space, doesn't move, and it's interpretable. I said, got us into humans. So, we're railing off really quickly because we got off to slow start. There are a lot of people I didn't mention, and there's a lot of people not on this slide. These are all the people I've had interfaced with over the years. I've been here 31 years now. I know a lot of people. I challenge anyone. I will buy you a beer if you can identify everyone on the slide. You can?
Yes, Doug Weber.
Everyone, not yes. Doug Weber. Doug Weber did his postdoc at Utah. He came to Utah and implanted dorsal ganglia with Utah arrays and did wonderful decoding work. And then he became famous. He became a program manager at DARPA for a brief period of time, and actually is the person who caused the haptic program to exist and work. But everyone. Who's Hudson? Kelly. Okay, so Kelly. I actually looked at this morning, and this person and this person, one is Sean Snow, and the other one is a different person. So yes, and this is, because I was giving this talk, I got a lot of emails from people or texts. Marshall Smith, anyone know Marshall Smith? He's a ENT doc. Also, he actually did recurrent laryngeal nerve implants with Utah arrays with Ryan McDermott, who's not on the slide. And not that I forgot about them, it's just bandwidth. And that is also a device that's just waiting to go to humans. That people who have surgery often have their laryngeal nerve severed. And if you could sense the other side, you could excite the muscle so that larynx works in tandem versus one side just flaccid and the other one isn't. Rick Rabbit asked, he's not up here. Holly is part of the Rick Rabbit group, but there's all sorts of people that do neural interfaces that are not up here. Okay, any questions? I apologize for, I hope my car's not towed away. I'll give you some recommendations if I'm allowed. Well, let me give my recommendation. So I said, I've been here 31 years. And for the life of me, I could not recover 31 years worth of stuff. All those pictures, I went to LinkedIn and got their current picture. And occasionally I found a picture that I dug through everything for the CNI website, even old versions pursued in Jake George. And you guys should save stuff. I talked about pizza, getting beer and pizza for being a booth representative. I had a picture of it sitting on the bulletin board, entering the was Norman, then Clark Warren, and now Dalrymple Lab. And of course, when they redid it for Ashley, that thing disappeared. Save these things. because someday you might be asked to do a talk about your experience. Okay,
So based on our,
sorry, I'm half deaf.
where do you see like neural interfaces going? Where's like the next
Ooh,
frontier
wow.
for it?
Can you repeat the question? Dave?
Oh, so the question, correct me if I'm wrong, where are neural interfaces going? Not down the toilet. That you saw, I mean, it took 30 years to get the vision stuff into humans and evaluating and finding out, yes, this is a viable idea. It took almost as long to get the auditory nerve work. It's taken a little less to get in peripheral nerve, but there's so many other things, so many other avenues that haven't been touched. Bladder control in the paralyzed. We've looked at that. It's feasible. There are issues, but this is the William Kolff answer, which is find the problem, identify it, identify the solution, move on. So that there are still lots of other things we can deal with. An obvious one is wireless stimulators, wireless recorders. So I'm going to New York next week to work with some people who are doing vagal nerve work. And they said, oh, well, I'll just put a connector subcutaneous. It's like, well, hold it. You got a 60 channel electrode array that you want to put a 60 pin connector subcutaneous. Whoa, that ain't gonna work. I've been there. So wireless, getting wireless done. However, that being said, second sight came up. There was a stimulator associated with second sight. Go try and get it. That the Al-Man Foundation has the rights to it, and they won't let you have that stimulator. There are research labs that have generated stimulators, but they're still not ready for prime time. And it is an issue between commercial product and research product. And if anything, I'll say we're moving more towards that. You need to think about this as a commercial product than as a research toy that I can gin up 5 of them in the lab, but geez, all the 5 of them won't be this like each other. So that's kind of been something that I've seen happening over the years.
Do you see a role of wireless electricity, or do you think like?
I am really still happening.
Oh, do you see a role of wireless electricity in going wireless, or do you think like the batteries are so small it's not, or so there's like?
That's a good question. So a lot of devices have an internal battery and you basically recharge it. Most of those are chest bone. That's a viable option. You run into a bandwidth issue, particularly and bandwidth equal power. If you're doing a lot of channel stimulation or recording or stimulation, excuse me, that so in the case in point, I'm thinking of the auditory nerve implant that we implanted. The device we put in was 15 electrodes, a 3x5 electrode, Utah slant array. And we could only attach 12 of them to the MED-EL stimulator because that's all they could do. And that, but it's all wireless. The stimulator has no battery. Pop that thing off and it's just a piece of inert stuff. And you can't say the same thing for a battery pack. So I think it depends on the application, probably. And hopefully, as you're doing that, you look to see what the market's, what's out there, what are people using, and how do we take advantage of what's available? Right?
Yeah.
Sorry.
So Neuralink, are you familiar with the work?
Who? Never heard of them. I almost threw that in, which is, so you know how Neuralink's implants? They've got an automated sewing machine, for lack of a better description. And this thing does it. And they can put in more electrodes than a Utah array. And as I said, remember a slide long ago, it needs a 25 by 25 grid. Well, the Utah array is 10 by 10. Where are you going to get the other array and a half? And Neuralink could potentially do that very readily. However, look at all the work that went into getting that device made, evaluated, certified, approved, et cetera. It's not something that's going to be done in a research lab. So, I wish them well. I mean, they're answering a lot of the basic questions that everyone in this field's been asking for. Remember that slide about the eye? It had a wireless system. Where's my wireless system? And where's my flying car? It's been forever. And Neuralinks, because Elon Musk has got piles of money and seems to get more money despite the fact he spends piles of money, that that's gonna be where it's gonna happen. Now, whether Neuralinks actually gets it to work, get perhaps a little bit of overambitiousness upon the owner's part. And that you need a little bit of more care. I don't know what the proper word is. That but that's you know, in the long run, that's helping. At least in my view. As long as you stop talking about making superhumans. Well. Yeah. Yes.
Sorry.
No, you weren't on any slides. Oh, you had, is that the question you had? All right.
What's your opinion on like biological development, that they're actually properly called? I was talking to one of my friends, mother who works for Medtronic. She works with pacemakers and electrophysiologists. And she was like, yeah, the next frontier is using organisms to do this rather than.
Oh, like organisms, things like that.
Yeah.
If it works, great.
Do you think that that's a possibility? Have you heard of anybody that you have a lot of respect for who's working in that kind of?
I haven't lived with deal that much.
Because I've never heard of it. No one would talk to me about it, and so I was surprised to hear that a private company would put something that's so far out in terms of technology.
Well, I mean, it was so far out, biological, organelles. That why build an artificial device when you could create your own artificial device? Why put in an artificial heart when you can grow it in pigs, transfer it over? That those are interesting ideas. And if they work, great. Biology is a rather complex problem. And that, no, it's great about bio. The reason I left my company, one, I liked the Utah job at Captain Park, and it was like, what I do. But because I was working on stuff in this fertility detection, and the life of me, I could look at what I was trying to do, and I could not train an AI to do it. You call AIs in those days, called expert systems, but I couldn't train a system to do something that an idiot like me could look at and go, oh, the problem is that meter is broken, that picture's wrong. Because the brain is amazing. And by extension, biology is amazing. Some of you have kids, right? When my wife was pregnant with my daughter, I was taking a neuro class in the School of Medicine, which was required for one of those days. And there's a whole bunch of stuff, how the doctors know is deficit. Things go bad. That's what they use to decide what's good. And all the bad things that can happen is terrible. There's an amazing that all of us are even alive. There are so many things happening, and this is a random world, and I'm surprised it works. I have a very nice, alive, thriving daughter, now 30, tried 30. So it worked, but to that end, it's like, can we recreate everything that's necessary to build a viable device and know what we need to do and know what we don't want to do? And that's a tough question. So that's kind of my long-winded answer on organelles and this perspective. But they provide us information. And the worst they can do is give us information. Go back to the golf experience. That weird thing, which is a big tub with a rotating disc that had one fluid on one side, blood on the other side. And a semi-permeable membrane between the two, and that's how they got rid of the urea. Just by the fusion. Just keep things moving. And a lot of people died before it got it working. A lot of animals died, but also a lot of people died. The Jarvik heart. That person died. But it provided the information to find out what's wrong. And there were a lot of animal experiments that went into the heart. It's called the Jarvik 7, it was probably a 1. And Jarvik wasn't the only one developing hearts at Utah. So a lot of things got found. And so you do stuff, you find stuff, and you decide whether it's fixable or not fixable. Any questions or something?
I think we better just do one more throughout the time.
I'll let you choose.
I just wanted to ask about the longevity of right, and especially for implantable devices. What's the longevity?
So that's a good question. Question was, what's longevity? So I will give you the brain pain experience. Everyone's worried about that. Yes, they put them in and they found failures, all like that. But they've had people implanted over a decade now, I believe, and they're still functioning. So that's, however, also there's the other study plan, which is what they do to make the Utah Array or any implanted device, A, last longer, not fall apart, integrated, B, not have an immune response. These are the things. So we're still carbide, that's the magic term for insulation. Here's work better. And also Patrick Cresco, and what we can do with it, because they know this equipment was happening, their immune response, what they can do at the right points actually reduce it. I think I the SBR that's got a really weird, interesting way of taking not a lot of talk about it. And it is that seems to have a laser effect on the immune response, and I'm a little worried. It's almost like magic, I don't believe in magic. So, let's see, that's this way you put it. And it turns out it is magic, but you can write magic. Then that would work. But applications, it turned out it really is magic. Then it's like, oh, wow. Fortunately, yeah. Yes. Thank you.